seeing tumors popping up really fast, and by 78 days, all of these guys have palpable lesions. If you treat them pretty early with this cross-linker, when you can inhibit the cross-linking in the stroma, you basically buy the mice, 20 days, which is pretty amazing considering how incredibly aggressive. And these mice, they're on this accelerated, so unfortunately, death sort of walk. And this is associated with the more pronounced reduction in the stiffness of the tissue. And not only do you reduce the actual or enhance the latency, but you also, the other guys that are non-treated, they grow pretty rapidly. These guys here actually are growing really slowly, and so Hamei's in the middle of doing a lot of histopathological analysis because we think that the actual histophenotype is probably altered. There's other things that are going on. She did some work to look at the actual metastatic foci, and I have to replace this particular graph because I think this is almost down to nothing here, right, Hamei? Yeah. So it actually almost obliterated the metastases, which is pretty striking. Possible mechanisms? So for the sake of our sanity here, we're not going to get into tons and tons of details. Suffice it to say that in the non-treated animals, just like in human, you have a lot of fibrosis. And when you inhibit the cross-linking, you reduce that fibrosis. So you can use markers. Here's like Picker series red. You can see that this is less red here, whether you use an inhibitor or an antibody inhibitory device. And the collagen is more relaxed, so you're basically inhibiting the cross-linking. What we are looking at and exploring in more detail has gone back to using this atomic force to begin to look at the invasive front of the lesions. I guess the coloration, I'm sorry, went off on this. But essentially what we were really surprised to find out is that the invasive front of where the tumors are actually invading out of, rather than being soft and where we would imagine MMPs will be functioning, is actually stiffer. So we're seeing these kind of stiffness kind of gradients. And this accords with work that John Condilas and other people have shown, where you see these fibrils of collagen that are actually emanating from here, plus you're going to have vasculature coming out, and that's where you see these epithelial cells dancing along. And so what we're trying to look at now is whether or not the rigidity is actually promoting the invasion and the migration, and how that might be driving sort of extravasation and metastases afterwards. That's something that we're actively pursuing. And the way that we're thinking about this, we started linked up with Joyce Wong, and then Jose Lopez is doing a lot of work here on this. It's a phenomenon called durotaxis, where cells will actually migrate towards a stiffer region. So this is kind of going completely contrary to the way that most people are thinking in terms of cancer metastases, where there's been a huge amount of money and effort put into MMPs. Most of you women here were conversant with a lot of the work that's been done on the MMP inhibitor trials that went out, and the clinical trials that actually had to be halted, with the idea that tumors actually made MMPs, and then they chewed up the matrix, and then the cells could invade, and that if you stop that, you could prevent metastases. Well, actually, a lot of the work that we're doing and others are now starting to do would suggest that, au contraire, basically the stiffness actually may be promoting the invasive behavior. And so if you inhibit the MMPs, probably it's a little bit simplistic to think that you're inhibiting stopping the softening and permitting the cells to invade in. So in terms of molecular mechanisms, there's a lot of ways to think about this. I'm going to just talk a little bit about cell adhesions, and I'll talk about a few other things to think about how this could happen. One of the things that we know is that integrins actually are one of the transducers that sense the extracellular matrix stiffness. They can interact with the matrix on the outside. They interact with and nucleate a whole lot of adhesion plaque proteins on the cytoplasmic phase. They interact with the cytoskeleton, and they signal through a whole host of signaling complexes. And so they're actually in a really good position to consider in terms of something that could be modified by mechanical force. We also think this is important because many years ago there were some studies that were done where you could remove normal cells, whether they were tumors or normal breast cells from a human or cell lines, and you could build these little organotypic structures. And you could take tumors, primary tumors or tumor lines, and you could show that they're really aberrant. And one of the big differences is they had altered integrins. And if you normalize those integrins, you normalize the behavior of these structures. And that held true for in vivo. And it was even though these guys had tons of mutations. So that would suggest that the integrins play a really critical role in driving sort of the expression of malignant behavior. And then a couple of years later, 2004, using transgenic models where you knocked out beta-1 integrin, they could show in this polyomino T model where you can see all the tumors that are coming up, but it's shocking how many tumors they have. Basically, they virtually reduced or obliterated all the tumors, and the few tumors that did come up are actually ones that the transgene was not properly knocked out. Okay, so how could cells potentially sense ECM stiffness through integrins? Well, you can imagine that cells have integrins on their cell surface. They're activated properly. We were able to show that it's nothing to do with the activation. They actually can nucleate and form adhesions in a really soft environment. And they do so, and we think that they're probably going to be quite dynamic. That's something that Brian is trying to look at in more detail. But for sure, the cells don't pull on the matrix a lot. So as I told you, the cells will tune their behavior, their viscoelastic properties to the actual stiffness of the matrix. And so in a soft matrix, they do very little force pulling. And this is like a little traction force assay that we use. And I'd be happy to explain to anybody who wants to know how to do that. Essentially, the blue is really low forces, pink is really high, and there are not a lot of forces here. As you begin to actually increase the cross-linking or the stiffening of the matrix, the cells are able to nucleate larger and larger adhesions, such that when they're in a really, really stiff environment, such as like in a cross-linked collagen in a tumor, what you end up with is you have these large signaling complexes that are formed, and the cells are actually able to exert a lot of tension on the matrix. And so there's this kind of reciprocal sort of interaction between the matrix and the signaling complexes, and then they exert more forces and they can actually remodel the matrix around them. And we think that's one of the ways that they actually use to remodel the matrix and allow themselves to make these little highways to invade. And you can, just show you that you can measure as there's an increase in the stiffness of the matrix, that there's an increase in the isometric force generation in these cells. Okay. And then this is just showing that it actually relates to the tumor, to the actual cell physiology. So on the soft gel, this is a marker of the integrin signaling. There's low signaling in the soft gel, and they don't spread a lot. In the stiff gel, they spread a lot more. There's a lot of that signaling going on, a lot of these adhesions that are formed, and you can see that measured here and quantified here. I think I'm getting close to being done. So how are you doing? Okay, good. So if you go back to the tumors and you look, just like human tumors, at experimental tumors, and this is not confined to breast only. This is also, we have some data from pancreatic cancers. The same thing happens in a lot of lung cancers. I think virtually every tumor that you look at has evidence of elevated signaling through these adhesion receptors. And in virtually every cancer, there's a change in the exocyte matrix. So it's not as though this is something that's peculiar to one form. Well, obviously, if you have leukemias, the matrix itself is not altered, but we don't know whether the behavior of the leukemia cells to mechanical cues is different. We think it could be. If you look here in the green, you can see there's a lot of FACT in these tumors. There's none virtually in a normal tissue, whether that's human or a mouse. We've done both. And if you inhibit the cross-linking, you can see that you basically obliterate or reduce that signaling through the integrins. So one of the things that, and this is like for those individuals that are doing experiments or thinking about this with respect to how can we relate this to what's going on in the clinic. So one of the things that matrix stiffness does is it modifies receptor signaling, not just integrins. Because many of us know that, basically tumors grow more and they invade and they do all kinds of bad things. And one of the things we all think about are things like EGF receptors, ERBB2 receptors and what have you. So what we're finding is, so this is just two pieces of data. One that shows that you can actually modify PGF receptor activation. So just modifying the stiffness of the matrix will change how the cells are able to activate and modify PGF receptors. So the autophosphorylation, or the fundamental signaling of the receptor, is changed, even with respect to basically its response to its growth factor. So it'll do something as basic as that, and that has relevance with respect to things like personalized medicine and therapies for personalized medicine. And in terms of signaling downstream of these receptors, in response to this, in this case, the growth factor called EGF, one of the quintessential signaling cascades that's activated is RAS and ERK. And what you see is in the soft matrix, there's a real little bit of RAS, or ERK signaling, whereas in the stiff matrix, the magnitude and the duration of that signaling is basically huge. So what you're doing is you're tuning the signaling downstream. Now in this case, it has nothing to do with the activation of the receptor, it's something downstream of the